Hello, welcome back to part 3 of our look at the immune and lymphatic systems. I wanted to just draw your attention here to this slide. This is a nice little summary table of things that we talked about during video 2. And so this talks about the lymph nodes and what the functions are. It talks about the spleen, talks about the mucosa-associated lymphatic tissue. Or the malt tissue, and it talks about the thymus as well. And so this is a nice little summary table for you to kind of check out and review as you are preparing for your exam. And so with that said, let's go ahead and let's jump into looking at video 3, which is talking largely about what we define as being the innate immune system. And so we left off at the last video with this idea that our immune response system has two major mechanisms by which it can prevent pathogens from causing some kind of infection or illness within our bodies. One of them being this idea of an innate immune system, and the other one being an adaptive immune system. And so video 3 here, this video that we're in now, is going to largely focus on the innate immune system. And so when we look at the immune system, as I just mentioned, we have this idea of this innate system, which is, by its very definition, the innate immune system is one that you are born with. And so you do not have to do anything. There is no developing of this. You are born with it. And so what constitutes the innate immune system? Well, your skin is part of the innate immune system. You're born with your skin. You don't have to do anything to acquire skin. And your skin is a barrier. It is made up of, in the epidermis, keratinized stratified squamous epithelium. And we know that that keratinization not only provides waterproof, but provides a mechanism of protection against things that are constantly being bombarded against that skin. And so it's preventing a whole slew of things from being able to enter into the skin. Now, in order to, because we know that the integumentary system, we know one of the functions of the skin is to help balance water, salt, and even remove some waste products. And so we do have these mucous membranes that are associated with the skin, in addition to sweat glands. And mucous membranes secrete mucus, and the idea is the purpose of that mucus is to trap and or slow down any kind of pathogen that may be trying to enter into the body. In addition to that, we also have internal defenses that are classified as part of the innate immune system. Again, this idea that you are born with some kind of natural protection system or mechanism that can try to slow down or fight off or prevent an illness or infection. And so what are we talking about? Well, we're talking about cells that are considered to be phagocytic. In other words, largely macrophages, and as we will learn, neutrophils. Right? NK cells, natural killer cells. Right? We'll talk more about them here in a few minutes. Inflammation. Yes, swelling. Inflammation is a mechanism that can prevent infection from spreading throughout the body. And so inflammation is actually a good thing. This is how we know healing is occurring. And of course, what we do is we naturally, immediately take an anti-inflammatory. Why? Because that swelling is uncomfortable, it restricts movement, it makes you feel achy, you can't do as much as you want to, and so it's annoying. And so we take an anti-inflammatory, which in a sense is shutting down your immune system. It's preventing your immune system from doing what it's designed to do. And so you will notice, and I'll say this numerous times, I very rarely take any kind of medication, especially anti-inflammatories. If I've got a swollen ankle, it's swollen for a reason. I need to sit down, keep it up, and let my body heal itself. And so there's reasons for that. Same thing with a fever. Fevers, it's a positive feedback mechanism, as is inflammation. And fevers are designed to kill off bacteria. And so bacterial proteins denature at approximately 101 degrees Fahrenheit, whereas human proteins denature about 105.6 degrees Fahrenheit. And so when you're running a fever of about 101 degrees, you're actually killing off bacteria, which is preventing the spread of a pathogen, right, or something that can cause illness. Same thing with inflammation. When you take an anti-inflammatory, you're now increasing the chance that that infection is going to spread throughout the body or spread to another area of the body. And so it does not make sense to go ahead and take that anti-inflammatory, especially without treating whatever the ailment is that is causing that inflammatory response. And then we have antimicrobial proteins as well. So they are your innate defenses, and we're going to go through each of these a little bit more in detail, especially the internal defenses here. We're going to talk a little bit more about neutrophils and how they do what they do. We're going to talk about natural killer cells and how they do what they do. They kind of are really a fascinating and interesting cell type, and so we're going to spend some time looking at that. We're going to look at the positive feedback mechanism that regulates inflammation. And so they're just some of the things that we are going to highlight. I would be remiss if I did not at least mention the adaptive immune system. Right? This is sometimes also referred to as acquired immunity. In other words, you're not born with this, but it develops as you are exposed to new pathogens. And as I have alluded to all along, your two main cell types that are functioning within acquired or adaptive immunity are your B cells, which is identified as being humoral identity. Why? Because it comes from bone. And the B cells, your humoral immunity, utilizes antibodies. And so your B cells are the cells that are actually producing antibodies within the body. T cells is cellular immunity, and the difference here is T cells actually use physical means to go ahead and kill off the pathogens. In other words, the T cells actually have to land on and dock, physical contact with those cells. And so these are basically what you're going to be looking at and what we're going to be discussing in videos 4, 5, and 6. And this video here is all about the innate immune system. And so let's go ahead and let's jump into looking at various aspects of the innate immune system. First thing, let's go ahead and define some cells, let's define some cell types that you are going to be responsible for. You'll notice not all of these are part of the innate immune system, and that is okay. It's good for you to know the differentiation between these guys starting now. And so, again, we have our NK cells, our natural killer cells. These guys are very closely related to cytotoxic cells. And so you might be asking, well, what is the difference then between a cytotoxic cell and a NK cell? And we're going to talk about that in a little bit. That is coming up. But as far as function goes, natural killer cells are kind of free-ranging. They're not specific. So in other words, they're not going to attack one specific cell. They're going to attack a broad range of cells. And so bacteria usually get destroyed by natural killer cells, or what I define as being NK cells. Cancerous cells are targeted by NK cells. Other infected cells, so a lot of times cells that are already infected with viruses become a target of natural killer cells, and we'll talk about why that is. Transplanted tissue. So if you have an organ transplant, you know that that individual is taking anti-rejection medicine for the rest of their life. Why is that? Well, that is because those anti-rejection medicines are actually inhibiting natural killer cells. They're blocking natural killer cells for being able to identify these other cell types. Now, you might sit there and say, well, okay, but that's kind of like a double-edged sword. And I would say, ding, 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 you are exactly correct. It is a double-edged sword. You have an organ that has failed. You have another individual's organ that is now transplanted into your body. They match it to a certain percent, but it's not a 100% match. And so your natural killer cells are going to identify the cells of that newly transplanted organ as being foreign, and it's going to attack it and destroy it. Just like it would a virally infected cell, or just like it would a cell that is cancerous and dividing uncontrollably, or just like it would attack a bacterium that should not be within the body. And so when you take anti-rejection medicine, you're shutting down the NK cells, which means you are now immunocompetent. Your immune system is compromised because you're missing a key player in the identification of virally infected cells. You're missing a key player in the identification and destruction of cancerous cells. And so now you're more prone to infections. You're more prone to cancer. You're more prone to bacterial infections when you're on those anti-rejection medicines. And so keep that in mind. Right? T cells and B cells, we've talked about them. I'm not going to spend much time on them now. You can kind of see there the premise of what you need to know and understand as far as why they are different right now. T cells are very specific for very specific antigens on a pathogenic cell. B cells using antibodies are very specific once again. All right. Macrophages, it's a generalist. It's part of your innate immune system. And so your macrophages are there to break down and consume and get rid of and clean up dead cells, damaged tissues, bacteria, and or they will very often work with T cells to identify foreign or pathogenic cells or cells and or tissues. And it will actually work with the T cells to go ahead and force what we would define as being apoptosis or programmed cell death. And then we have these dendritic cells, which very often are alertist. And so they will alert T cells and B cells, as well as other cells, to the presence of pathogenic cells. And so we find very often these dendritic cells within the epidermis. We find them within the mucous membranes. And we already talked about the fact that we find them in the lymphatic organs, primarily the thymus and your secondary organs, your secondary lymphatic organs. And so I'm just going to kind of quickly go through this and kind of review with you the leukocytes. We're going to spend a little bit of time on neutrophils because they are very interesting. This is a neutrophil right here. Actually tracking down a bacterium that you see right there. And you will eventually see that this little guy right here basically engulfs the bacterium. There it is right there. It's now inside of the neutrophil. And it will go ahead and cause that bacterium to break down. Let me play that again for you. It might not let me. Um, I'll replay that for you here once we are done. But interesting as it is, neutrophils play a wide range of roles and functions. So, uh, neutrophils very often are found in connective tissues. We also find them within the lymph nodes. And they have two primary ways of destroying the bacteria: phagocytosis, which is what you just witnessed here. Let me kind of come back down here. Play that again for you so you can see again the bacterium is right there. There's another one right there. All right, that neutrophil is coming around, catching up to him, zigs and zags, pushing through the red blood cells that you see there, and there you go. He has now engulfed that bacterium. And at this point in time, he will basically secrete enzymes that will force that bacterium into apoptosis and lead to the destruction of that bacteria, once again through phagocytosis. And then we have this idea of a killing zone. Now, here's what's unique about the killing zone. The neutrophil basically goes kamikaze. In other words, it's going to sacrifice itself and its own destruction for the destruction of hopefully the pathogenic cell. And how does it do that? Well, the neutrophil will go ahead and it secretes or releases these little enzymes called lysozymes. All right, and these lysozymes, is another S in here. Let me rewrite that for you. These lysozymes force the release of enzymes. And think of the enzymes as being almost like this nuclear bomb that is going off. It gets released into the interstitial fluid. Okay, once again, remember we're primarily focused in connective tissues here. And so you're talking about things like the dermis of the skin. You're talking about things like um, dense irregular and dense regular connective tissue. You're talking about reticular tissue. All of your connective tissues, these enzymes get released from the lysozymes into the interstitial fluid. And what they do is the enzymes rapidly cause the absorption of oxygen. And so it sucks the oxygen out of that interstitial fluid. And when it does that, it sucks into the enzyme the oxygen and it adds an extra electron onto the oxygen. And it creates something that is called a superoxide ion. It is an oxygen molecule that has an additional electron that has been added onto it. And so it gets this little asterisk up here. And we call that a superoxide ion. Now, why is that important for you to understand? Well, because the superoxide ion, this O2 molecule with this extra ion or this extra electron, reacts with hydrogen ions. It's going to react with a hydrogen ion because remember now it wants to try to balance this extra electron out. And so it reacts with O2 superoxide plus a hydrogen ion, right? Actually 2 hydrogen ions, is going to equal H2O2, which is hydrogen peroxide. And hydrogen peroxide is highly toxic to cells. And so it's actually forming this gaseous cloud of hydrogen peroxide and something that is referred to as hypochlorite. And so both of these together, the hydrogen peroxide and the hypochlorite, form this gaseous cloud that is referred to as a killing zone. And what ends up happening is anything that comes in contact with this killing zone, with this gaseous cloud of hydrogen peroxide, or I'm sorry, with hydrogen peroxide and hypochlorite, basically becomes destroyed. Whether it's a good cell, whether it's a bad cell, whatever it is, and the neutrophil is also destroyed as well. And so again, that neutrophil is sacrificing its own self for the, if you would, the betterment of the rest of the body and the organism. And so fascinating cell type. Here we have a neutrophil, this white blood cell, this leukocyte, that has the ability to stimulate T cells and B cells, as we've already talked about. It has the ability to go ahead and induce clotting. It has the ability to do phagocytosis. It has the ability to form this gaseous cloud around it that will eliminate and destroy and basically force into apoptosis good cells, bad cells, and itself. This is why neutrophils are so plentiful throughout the body. Again, you have eosinophils and basophils. All right, we know that eosinophils are leukocytes that protect against both parasitic infections and allergens, and so they cause inflammation. All right, they cause inflammation. Basophils, we know, are cells that release histamine and heparin in response to, once again, very typically either allergic reactions or, in the case of mast cells, remember, basophils that have migrated into connective tissue, such as the dermis, become what we also define as being mast cells. And so mast cells have the ability to go ahead and once again cause the release of histamine and heparin. Now think about this. Heparin is a blood thinner. Histamine is a vasodilator and a bronchial constrictor. And so it constricts the bronchi to prevent anything from getting into the lungs because the lungs are super sensitive. The lungs are a dark, warm, moist environment. That is what we call an incubator for bacteria. All right. And so it wants to go ahead and that histamine causes that bronchial constriction to try to prevent something from getting down into that environment that's going to become a breeding ground. At the same time, it's a vasodilator because it wants to open up that blood flow to try to help flush out whatever it is that's getting into the system. All right. So what do? Well, we go ahead and we take an antihistamine, which again shuts down your basophils. It shuts down the immune response. And so it prevents bronchial constriction, which means now you are more prone to some kind of upper respiratory or lower respiratory infection. It does not allow for vasodilation. And so that means that your eyes are not watering as much, your nose is not running as much to try to go ahead and eliminate whatever that allergen is within your body. So it really is counterproductive. The other thing is heparin as a blood thinner prevents blood clotting. And so this is why mast cells largely secrete it within the dermis because it prevents that blood clot from forming. It flushes the wound, and so it actually encourages bleeding, or it encourages, in the sense of, in the case of basophils, it encourages the blood to flow quicker, to go ahead and try to, again, get to the mucous membranes so it can be flushed, whatever that allergen is that is wreaking havoc. All right, and again, we have lymphocytes, specifically natural killer cells. We know T cells and B cells are not part of the innate immune system, but they are a type of lymphocyte, so I did list them there. But natural killer cells are a type of lymphocyte. That is a type of lymphocyte, and so I do list that there. And then we have monocytes as well. All right. What else do we have, though, to go ahead and try to prevent an infection that is part of our innate immune system? Well, we've got these other things here that are referred to as interferons. And so an interferon is actually a enzyme. It's a protein that is released from cells that have been infected with a virus. And so an infected cell by a virus releases the interferons, and these interferons diffuse into nearby cells, and the whole point is to block viral replication. So the infected cell is done. The virus has gotten in. It's going to release its genetic components, whether it's DNA or RNA, into that infected cell. The cell is going to go ahead and replicate the viral parts, and it's going to release these little virions, these newly formed viruses that have not yet infected a cell. And their first task is to find a newly uninfected cell, which is going to be a cell that's nearby. And so what ends up happening is the interferons go into the healthy cells, bind to receptors that triggers the DNA to produce antiviral proteins that will block that viral replication. It's a very cool system. Here it is right here that you're looking at. And so here comes the virus. It's coming in. It's releasing its DNA, which is going into the host cell's DNA, and it's leading to the viral replication of new viruses. But at the same time, the gene for the interferon is turned on. Interferon is now secreted. It binds to the interferon receptors on the cell membranes of nearby cells, and it's what we call a second messenger system. And so the interferon binds to the receptor on the cell membrane that triggers a response in the cell. And that response is to activate a gene on the chromosome in the host cell to create an antiviral messenger RNA, or an antiviral protein that will inhibit, here's your antiviral protein right here, that will inhibit a virus that infects from going ahead and replicating. So it blocks that viral replication. So it's a very cool mechanism that we have for preventing, or I guess the better term is slowing down, the rate of viral infection. It's a roadblock. It's not an end-all or be-all, because it's not 100% effective, but it will slow down the rate of viral replication. And so that buys the adaptive immune system some time to get up to speed, which is what you want. The other thing is the complement system, which actually causes cytolysis of microbes. And what we mean by that is it forces those microbes, whether it's bacteria, whether it's a pathogen, whether it's an allergen, whatever it might be, it forces those cells into apoptosis. And so the cell membrane ends up rupturing at that point in time. And then we have natural killer cells. And this is actually pretty much where we're going to stop. We're going to kind of review this idea of the natural killer cells. And I apologize, this video is a little bit longer, but that's okay. And so natural killer cells they make up about 5 to 10% of the cells within the blood. They belong to the lymphocyte family. Right? We find them also in the spleen. We find them in lymph nodes. And they will literally attack any cell that is not normally found within the body. And what they're doing is they're looking for a lack of or a different antigen. And so if a cell does not have an antigen that is identified as self or belongs to you, or if the cell is lacking any kind of antigens altogether, natural killer cells will go ahead and basically attack. And when they attack, what they do is they force the identified cell into apoptosis. And they do that by causing swelling within the cell. And so what we want to do is we want to look at that process that is involved in natural killer cells leading to the destruction of a cell. Before we do that, I want to introduce to you a couple terms that we're going to be picked up with later on. As we start looking at T cells and B cells. So all cells have a receptor on their cell membranes that are called MHC1 proteins. All right. And these are receptors that will basically bind to antigens that it comes across to see if it is part of or not part of the identified self. All right. Cells that lack MHC1 proteins, natural killer cells attack. All right. And so if the cell does not have MHC1 proteins, natural killer cells say, you don't belong here. You're not part of me. You're not human. And it will attack it and cause and go through this process that we're going to look at, forcing it into apoptosis. Cells that are coated with antibodies. All right. So if a cell is covered with antibodies, then the natural killer cell says, nope, you don't belong here because you're covered in antibodies. And only cells that are pathogenic get covered in antibodies. And so it will go ahead and it will attack those cells as well. Basically, antibodies mark cells for destruction, and the natural killer cells detect that. Or if a cell, so these proteins, these receptors on the cell membrane, when the cell undergoes stress, very often those proteins, those receptors, change shape. They change shape due to stress. Right? Cancerous cells do that. They're rapidly dividing, and that causes stress to the cells, which changes the protein structure of these receptors. Natural killer cells can detect that. And when they detect that, they go ahead and they attack and destroy those cells. And so natural killer cells have these ways, these mechanisms of overcoming their generality to make sure that they don't attack cells that are of self. Right? Again, they're going to look for a lack of MHC1 proteins. MHC stands for major histocompatibility proteins. Right? So major histocompatibility 1 proteins. Cell doesn't have that. Natural killer cell says, nope, you don't belong here, marks it for destruction and destroys it. It's going to look for cells that are covered in antibodies, because antibodies mark cells that don't belong there for destruction. It's going to take them out. All right? And the third thing it's going to look for is when cells are stressed that do belong to you, their receptor proteins, their membrane markers change shape. And so it's looking for cells that are stressed because very often they are cancerous cells. And so the natural killer cells are going to go ahead and take that out as well. And so there's another type of cell that is referred to as a cytotoxic T cell. A cytotoxic T cell. You're going to see it abbreviated TC cells, cytotoxic T cells. These T cells actually function the same way as natural killer cells do. And they use pretty much the same mechanisms for destruction as natural killer cells do. The difference is T cells are very specific and they target very specific cells. And so they're not generalist, but they use the same mechanism for destruction. And we're going to talk about that here in a few seconds. So what is this process that natural killer cells use? Well, here's our NK cell, right? And it has bound itself to an identified cell that is not of self, right? Maybe this is a cancerous cell. Maybe this is a cell that is covered in antibodies. Maybe this is a cell that is lacking MHC1 proteins. Right? Whatever it is, NK binds to it. And what it does is it will secrete an enzyme called perforin. And perforin perforates, it puts a hole into the membrane of this marked cell. Then the NK cell secretes an enzyme called granzymes. And the granzymes flow through the hole that the perforin proteins have created. And once inside, the granzymes force that cell into apoptosis. All of this extracellular fluid flows in, and then the cell ruptures and dies. It undergoes programmed cell death. It's considered programmed cell death or apoptosis because the granzymes are driving the destruction of the cell. It's actually taking over the genetic makeup of the cell and forcing it into destruction. And then a macrophage comes by and will go ahead and engulf or phagocytize what we refer to as endocytosis. It brings it into the macrophage and it will go ahead and continue the breakdown and the destruction of that targeted cell. We've kind of already talked about inflammation and fever a little bit, and so I'm not going to spend too much time on there. You can kind of look at this, again, symptoms of inflammation: redness, heat, pain, and swelling. The redness and the heat comes from influx or a rushing in of blood, and then kind of seals off that area. And again, the purpose is to get rid of any kind of microbe or toxin, or to prevent the spread of infection. So inflammation is going to seal off an area so things can't get in, and it's also not going to allow other things to escape. And again, fevers can go ahead and intensify this idea of an interferon. And we can see that the trigger is bacterial toxins, what we refer to as endotoxins, or toxins that have been released from the bacteria. And the process of inflammatory response, you can see here we have the initial injury that has occurred. Right? And so let's say you have tissue injury, because remember, inflammation is always going to happen within the tissues. All right, and so histamine gets released. Kinins will get released. Kinins are enzymes that are typically secreted from bacteria that will go ahead and usually initiate or at least alert that there is damage to cells in the nearby. Prostaglandins. Prostaglandins will increase histamine. Prostaglandins is a type of steroid that acts locally. And so it is what we refer to as a paracrine. Right? Paracrine are hormones that are released from a cell and stays locally to affect cells within the given area. And so as part of the damage to the cells, histamine is released typically by mast cells. Complement mechanisms take over, right, such as fever, histamine again being released, heparin being released, kinins being released from within the damaged cells or the bacterial cells, and this hormone prostaglandin that's going to intensify the inflammatory response. Um, and so you can see because of this, all right, arterioles are going to dilate. We know that that is an impact of histamine. All right, increased blood flow into the area, which is going to allow for that increased blood flow into the area is going to increase the heat and the redness. All right, and so that's going to increase the metabolic rate of those cells in there. Why? To try to speed up the production of interstitial fluid to go ahead and increase the swelling. Increased capillary permeability. Right? Again, this is what's going to go ahead and drive the production of interstitial fluid, right? Which again is going to cause swelling, which is going to cause pain and swelling. Right? That's what's going to immobilize the area. That's what's going to cause the stiffness. Think of it as a water cast that's being created from inside the body. Right? And then again, neutrophils, monocytes, lymphocytes are all going to come on in. And when that happens, right, leukocytosis, in other words, white blood cell proliferation is going to go on. White blood cells are going to come into the area, and that's going to go ahead and initiate the cleanup, the attacking of, the destruction of any kind of cell that does not belong there. So whether it's damaged tissue, whether it's bacteria, whether it's viral, whatever it might be that might be causing the initial damage to the tissue will go ahead and be taken care of. And then again, all of that should lead, you've pushed the body way out of homeostasis, but all of that should lead to healing within that tissue. And so we are going to kind of stop there for right now. There's a lot for you to take in and digest with this video. And so I want to give you that time to be able to do that. And so take your time, go back over, rewatch this a couple times, and I'll catch you on the flip side.